We are launching into a deep exploration of the fundamental link between a molecule's intrinsic properties and its capacity to penetrate biological barriers, which we define as drug absorption. This analysis centers on how physical parameters, including particle size and polymorphism, intersect with chemical attributes like the pKa and partition coefficient to govern the movement of a substance into the bloodstream. View these physicochemical factors as the precise molecular criteria required for a drug to traverse complex lipid bilayers. Understanding these mechanics allows us to predict and control exactly how effectively a therapeutic compound will reach its target site.
Bioavailability stands as a rigorous metric defining both the rate, how quickly a substance traverses membranes, and the extent, the total percentage of the dose reaching systemic circulation intact. Biopharmaceutics is the core science examining the dynamic tension between a drug's physicochemical identity, its formulation architecture, and the specific route of administration. Analyze how a change in the dosage form might drastically alter the absorption velocity, while the total amount absorbed remains static. Our goal here is to quantify exactly how these three variables dictate the final concentration profile within the blood.
Look closely at this kinetic sequence that dictates exactly how a pharmaceutical compound transitions from its solid state to a functional molecule within the body. The process initiates with disintegration into discrete particles, which must then undergo dissolution to become a solute in the gastrointestinal environment. This dissolved state is mandatory for the final phase, absorption, where the drug permeates the biological membranes. Pay special attention to the bottleneck in this series. The slowest kinetic phase is defined as the rate-limiting step that ultimately governs the overall velocity of systemic bioavailability. Observe how the chemical nature of a molecule dictates which specific vector in this sequence acts as the primary kinetic bottleneck. For lipophilic substances such as cresofulvin, the extremely low aqueous solubility causes the transition from solid particles to a dissolved solute to be the slowest phase, making it a dissolution rate-limited process. Conversely, if a drug is hydrophilic, it dissolves rapidly but struggles to penetrate the lipid-rich biomembrane, shifting the rate-determining step to the permeation stage. You must distinguish between these two distinct hurdles because the slowest individual segment in this molecular relay determines the absolute velocity and concentration of the drug reaching the systemic circulation. Focus your attention on hydrophilic molecules such as cromolyn sodium, which transition into a dissolved state with high velocity, but then encounter a major kinetic obstacle at the biological membrane. Because these molecules favor aqueous environments, they struggle to diffuse through the hydrophilic lipid bilayer, shifting the rate-determining step to this final stage of permeation. Examine the flowchart where the primary bottleneck is explicitly labeled as transmembrane rate-limited for these substances. Essentially, the total speed at which the drug enters the body is not dictated by how fast it dissolves, but remains strictly governed by the resistance it faces during this membrane crossing. Observe this roadmap of 8 physicochemical parameters that systematically determine whether a molecule successfully reaches the systemic circulation or fails at the biological threshold. We prioritize the pH solubility profile and lipophilicity because they govern the equilibrium between the drug in solution and its subsequent ability to permeate the lipid bilayer. Analyze how structural variables like particle size, polymorphism, and hydration state directly manipulate the surface energy and kinetic velocity available for dissolution. Furthermore, we must evaluate how pH stability, complexation, and chemical modifications act as final determinants of the molecule's chemical integrity and effective concentration at the site of absorption. Look at this graph where we plot drug concentration in micrograms per milliliter on the vertical y-axis against a range of physiological pH values on the horizontal x-axis. This pH solubility profile is our primary tool for predicting the total dissolution potential of a dose as it transitions through different segments of the gastrointestinal tract. Observe the specific curve for glyburide. The solubility is restricted to a very low baseline in the acidic stomach environment, but undergoes a massive exponential increase as the pH rises toward the intestinal range. This sharp upward trajectory tells us that the molecule's ability to enter the solution is critically dependent on reaching a more alkaline environment. Analyzing these plots ensures we can calculate exactly where and when a drug will be available for subsequent absorption into the bloodstream.
The pH-partition hypothesis establishes a rigid framework where drug molecules must cross biological barriers primarily via passive diffusion, moving from a region of high concentration in the gastrointestinal tract to the bloodstream. Observe the interplay between the ionized and unionized particles depicted in the diagram. The presence of an electrical charge significantly increases a molecule's polarity, making it incompatible with the hydrophobic lipid membrane. This theory posits that the unionized, neutral form of a drug is the only version capable of partitioning into the lipid bilayer effectively. Therefore, the total rate of absorption depends entirely on the fraction of the drug that remains uncharged, which is itself a direct consequence of the surrounding pH and the molecule's own pKa.
Mastering the Henderson-Hasselbalch equations allows us to calculate the precise ratio between a drug's ionized and unionized forms. For weak acids, the pKa minus pH value is equal to the logarithm of the unionized concentration divided by the ionized concentration, which explains why an acidic environment shifts the equilibrium toward the neutral form. Shift your analysis to weak bases, where the same pKa minus pH calculation equals the log of the ionized concentration over the unionized concentration. Note the critical inversion of the ratio here. Solving these logarithmic relationships provides the exact percentage of drug capable of crossing the lipid membrane at any specific physiological site.
Analyze the vertical gradient of this chart, which ranks chemical species according to their pKa, the negative base 10 logarithm of the acid dissociation constant. Notice that as the pKa value decreases into the negative range, such as hydroiodic acid at -10, the proton donating capability hits its maximum, identifying the strongest acids at the top. Conversely, the inverse correlation dictates that the weakest acids, found at the bottom with pKa values as high as 50, correspond directly to the most potent, strongest conjugate bases. Trace this diagonal relationship to see how the mathematical magnitude of the pKa serves as a precise predictor for the equilibrium position of any acid-base reaction.
This table integrates the Henderson-Hasselbalch equations to categorize drugs based on how their chemical strength dictates membrane permeability. For acids, you calculate the log of the ionized over the unionized concentration by subtracting pH from pKa, while for bases, this exact calculation yields the log of the ionized over the unionized concentration. Notice that very weak species, such as acids with high pKa or bases with low pKa, stay unionized regardless of the environment, making their rapid absorption entirely independent of gastrointestinal pH. At the opposite end, strong acids and bases remain ionized across the entire pH range, which creates a permanent barrier to absorption. The most critical group involves substances with moderate pKa values, where weak acids are better absorbed in the stomach because pH is lower than pKa, while weak bases shift toward the unionized form in the intestine as pH rises above pKa.
Analyze this fundamental breakdown of the pH-partition theory, where physiological mechanics overrides simple chemical equilibria. By applying the Henderson-Hasselbalch relationship to aspirin with a pKa of 3.5, we find that while 99% is unionized in the acidic stomach at pH 1.0, this neutral fraction drops to a negligible 0.1% in the small intestine at pH 6.5. This presents a paradox. Why does most absorption occur in the intestine despite such an unfavorable ionization ratio? The answer lies in two physiological compensators, the vast epithelial surface area and the extended residence time of the small intestine, which provides significantly higher total flux than the stomach environment. You must conclude that while chemical state is important, the physical architecture of the absorption site is the ultimate determinant of systemic uptake.
Look at these critical exceptions where the pH partition theory fails to predict molecular behavior. Consider quaternary ammonium compounds. Despite maintaining a permanent positive charge across the entire physiological pH range, they still successfully traverse the gastrointestinal barrier.
This occurs because charged molecules can engage in ion pairing, where they associate with oppositely charged organic ions to form a net neutral complex capable of partitioning into the lipid membrane. Furthermore, the theory focuses exclusively on passive diffusion, but you must account for active transport mechanisms, where specific protein carriers facilitate the movement of ionized molecules against their concentration gradient. How do these bypass mechanisms fundamentally challenge our reliance on the Henderson-Hasselbalch ratio as the sole predictor of absorption?
We must confront the reality that being unionized is not a guaranteed ticket through the biological membrane, as many compounds exhibit poor absorption even when their neutral form predominates. This discrepancy arises because the pH-partition theory fails to account for the physical dimensions of the drug, specifically the molecular weight. A high molecular weight creates a significant kinetic barrier, where the sheer bulk of the molecule results in low permeability regardless of its charge. When analyzing a drug's potential, you must integrate its structural volume into your calculations, recognizing that a massive molecular skeleton will struggle to diffuse through the dense lipid bilayer. What happens to our absorption models when the size of the molecule overrides its chemical state? Analyze the discrepancy between barbitone with a pKa of 7.8 and thiopentone at 7.6, which both remain almost entirely unionized in the stomach's acidic environment. According to the pH-partition theory, their absorption should be nearly identical, yet thiopentone penetrates the gastric barrier at a significantly higher rate. This occurs because thiopentone possesses a much higher partition coefficient, signifying an elevated lipid solubility and a stronger chemical affinity for the gastrointestinal membrane compared to barbitone. You are witnessing a critical limitation of the theory. Once the molecule is neutral, its inherent lipophilicity becomes the dominant force driving it into the lipid bilayer. Therefore, you must evaluate the partition coefficient as an independent variable that can override similarities in pKa and ionization status. Even when a molecule achieves the ideal unionized state, absorption remains stalled if the substance lacks sufficient lipophilicity to partition into the biological barrier. We quantify this lipid solubility using the oil-to-water partition coefficient, designated as the variable K, which represents the mathematical ratio of a drug's concentration in a lipid phase versus its concentration in an aqueous phase at equilibrium. Examine the direct proportionality shown here. As the numerical value of K rises, the molecule's chemical affinity for the hydrophobic lipid membrane increases accordingly. This relationship dictates that a high K value serves as the essential thermodynamic driver, forcing a dissolved drug out of the aqueous gastrointestinal fluid and into the lipid-rich core of the cellular membrane. This dataset provides a quantitative validation of how lipophilicity directly dictates systemic uptake. Compare the partition coefficient, expressed as the ratio of drug concentration in heptane versus water, for thiopental at a high 3.3 against its 67% absorption rate. Then contrast this with barbituric acid, where a partition coefficient plummeting below 0.002 results in a mere 5% absorption. The mathematical relationship here is unambiguous. As the numerical value of the partition coefficient increases, the efficiency of membrane penetration follows a corresponding upward trajectory. You are looking at a clear hierarchy, where the magnitude of this ratio acts as a primary lever for determining whether a drug's absorption is classified as rapid, moderate, or slow. We must analyze the pH stability profile, which mathematically plots the reaction rate constant for drug degradation on the y-axis against the physiological pH range on the x-axis. Notice how erythromycin exhibits extreme sensitivity to hydrogen ion concentration. Its degradation rate constant spikes in the acidic environment of the stomach, indicating rapid molecular decomposition. Conversely, as we move toward neutral or alkaline pH values, this reaction rate constant drops significantly, demonstrating that the molecule achieves kinetic stability only in higher pH environments. To bypass this acidic instability, we either apply an enteric coating to shield the tablet from gastric fluid, or utilize a prodrug like erythromycin stearate, which chemically alters the molecule to resist degradation until it reaches the absorption site. How does this shift in the reaction rate constant fundamentally change our choice of dosage form design?
Analyze this linear kinetic sequence, where reducing particle size serves as the primary mechanism to expand effective surface area, directly increasing the velocity of dissolution and subsequent absorption. By implementing micronization, we can achieve such high bioavailability that the required therapeutic dose for molecules like spironolactone is mathematically reduced to just 1/20th of its original mass. You must account for the high surface energy of ultrafine particles. Specifically, when diameters fall below the 100 nanometer threshold, spontaneous reaggregation occurs, which paradoxically collapses the surface area. Reflect on the strategic exception where slow dissolution is intentionally maintained for gastric irritants, proving that chemical stability and patient tolerance sometimes override the drive for maximum absorption speed.
Decreasing particle size to the micron level creates a massive theoretical surface area. Yet for hydrophobic substances, this physical change alone fails because the particles inherently resist contact with aqueous fluids. You must overcome this resistance by introducing wetting agents, such as sodium lauryl sulfate or tweens, which lower the interfacial tension between the solid drug and the gastric medium. Without this reduction in tension, the fluid cannot displace the air surrounding the particles, effectively preventing the liquid from wetting the surface for dissolution to occur. Within the body, biological surface active agents like bile salts perform this exact chemical function, facilitating the transition of poorly soluble drugs into a dissolved state within the small intestine. Ask yourself, if the liquid cannot touch the surface of a particle due to hydrophobic repulsion, does the total surface area even matter for the final dissolution rate? Examine how the internal architecture of a solid drug dictates its ultimate fate in solution by distinguishing between crystalline and amorphous forms. While a crystalline lattice provides stability, its high lattice energy results in much lower aqueous solubility compared to the disordered, high-energy state of an amorphous form. For instance, the amorphous form of novobiosin is precisely 10 times more soluble than its crystalline counterpart, demonstrating how removing the rigid molecular framework drastically lowers the energy barrier for dissolution. Even with an identical chemical formula, such as in chloramphenicol, these distinct spatial arrangements, or polymorphs, exhibit vastly different physical properties, including density, hardness, and compression characteristics. Which specific molecular arrangement would you prioritize if your primary goal was to bypass a dissolution rate-limited absorption step? Examine this kinetic profile where we plot serum concentration in milligrams per liter on the vertical axis against a 12-hour timescale on the horizontal axis to compare the performance of different chloramphenicol polymorphs. Notice that Form B achieves a peak concentration nearly double that of Form A, while a 50-50 mixture results in a precisely intermediate bioavailability curve. This dramatic shift occurs because polymorph B exists in a higher energy state with a less stable crystalline lattice, which mathematically translates to a higher dissolution rate compared to the more stable, low-energy lattice of Form A. By simply altering the internal crystalline arrangement without changing the chemical formula, we can manipulate the maximum concentration achieved in the blood. If you needed to double the effective concentration of this antibiotic without increasing the dose, which specific polymorph would you incorporate into your formulation? Hydrates occur when water molecules become integral parts of a drug's crystal lattice, forming what we call solvates. For organic compounds like ampicillin, the anhydrous form typically exhibits significantly higher solubility because it lacks the stabilizing water molecules that lower the free energy of the hydrated crystal. Study the provided graph, where the concentration of ampicillin is measured over a 60-minute interval. The anhydrous preparation achieves a steady-state concentration of 10 milligrams per milliliter. In stark contrast, the trihydrate form, which incorporates 3 water molecules into its structure, plateaus at a much lower 8 milligrams per milliliter. This data confirms that the hydration state acts as a fundamental physical barrier to reaching maximum saturation. If you were designing a dosage form where rapid absorption is critical, why would you choose the anhydrous form over the trihydrate? Complexation occurs when a drug molecule coordinates with a ligand, which can be an endogenous substance, a dietary element, or a formulation excipient. Specifically, analyze how mucin within the intestinal mucus binds to streptomycin and dihydrostreptomycin, creating a molecular association that sequesters the drug and prevents it from interacting with the absorption surface.
In the small intestine, bile salts can also react with neomycin or kanamycin to form precipitates that are chemically insoluble and thus entirely non-absorbable. This formation of large, stable complexes fundamentally alters the drug's physical state, rendering it unable to penetrate the lipid bilayer. If these biological components are effectively locking the drug in a non-absorbable state, what formulation strategies could you employ to shield the molecule from such interactions during transit?
Consider the chemical trap that occurs when a drug molecule binds with external ligands, a process we define as complexation. When tetracycline encounters divalent cations like calcium, it undergoes a chelation reaction, resulting in a molecular structure that is entirely insoluble and thus incapable of membrane transport. This same inhibitory mechanism occurs when specific excipients are introduced into the formulation. For instance, utilizing dicalcium phosphate as a filler or pairing phenobarbital with polyethylene glycol 4000 triggers the formation of non-absorbable precipitates. By forming these large, stable aggregates, the drug is effectively sequestered in the gastrointestinal lumen, rendering it biounavailable despite being physically present in the dose. How must we reevaluate our choice of fillers and dietary instructions to prevent these irreversible chemical locks from forming during transit?
Cyclodextrins represent a sophisticated class of enzymatically modified starches that organize glucose units into precise ring structures containing 6, 7, or 8 units, designated as alpha, beta, and gamma variants. This circular architecture establishes a critical dual polarity environment, where the outer surface is entirely hydrophilic, while the internal core maintains a hydrophobic cavity. Lipophilic drug molecules can then be sequestered within this central void to form a soluble inclusion complex. By shielding a water-insoluble drug inside this molecular host, the entire complex achieves high solubility in the aqueous gastrointestinal environment. How does this specific physical encapsulation fundamentally alter the concentration gradient at the biological membrane compared to a free, insoluble drug particle?
Micronization serves as a classic example of a drug whose therapeutic potential is restricted by its inability to dissolve in aqueous environments, leading to poor oral bioavailability. We overcome this hurdle by utilizing cyclodextrin as an excipient to form a soluble inclusion complex. Look at the diagram. The hydrophobic micronization molecule enters the internal hydrophobic cavity of the cyclodextrin, while the outer hydrophilic surface remains exposed to the surrounding water. This specific molecular arrangement shields the drug from the aqueous environment, which mathematically increases its effective solubility and dissolution rate. By strategically manipulating these intermolecular forces, how much more effectively can we deliver a lipophilic drug to the systemic circulation? We reach our final parameter, where we go beyond formulation and actually alter the drug's molecular structure to optimize performance. By chemically modifying tetracycline into the derivative doxycycline, we successfully engineer a molecule with significantly higher lipid solubility. This structural shift ensures that doxycycline partitions more readily into the intestinal membrane, which drastically increases the efficiency of its systemic absorption compared to its parent compound. Crucially, this modification also reduces the molecule's chemical affinity for calcium ions, effectively preventing the formation of those non-absorbable precipitates that previously stalled bioavailability. How does this dual improvement in both lipophilicity and stability against chelation redefine our ability to predict the therapeutic success of an oral dose?
Assess the kinetic divergence between tolbutamide as a free acid and its chemically modified disodium salt. Chemical modification through salt formation accelerates the dissolution rate by a factor of 5,000 at an acidic pH of 1.2 and by 275 times at a physiological pH of 7.4, showcasing a massive shift in the molecule's affinity for the aqueous medium. Witness the resulting blood sugar profile, where the salt's rapid dissolution causes an abrupt drop to 70% of the baseline, a response that is often too volatile for clinical safety. Clinically, the free acid form is preferred because its slower, controlled dissolution velocity facilitates a more gradual and sustained therapeutic effect over the entire 10-hour observation window. Could you justify intentionally selecting a molecule with a 5,000-fold slower dissolution rate when our general objective is usually to maximize the speed of absorption?
Observe the dramatic variation in the dissolution velocity of penicillin V when we manipulate its chemical identity through different salt forms. Notice how the potassium salt achieves an 80% dissolution within 12 hours, representing a high-energy transition into the aqueous state that favors rapid oral absorption. Conversely, the benzothine salt displays an intentionally depressed kinetic curve, reaching only 20% dissolution in that same timeframe. This slow-release profile is the exact scientific requirement for a depot injection, where a stable, long-term concentration is necessary rather than a sudden spike. Which molecular modification would you select if the clinical goal was to maintain therapeutic levels over several days rather than a few hours? The Biopharmaceuticals Classification System provides a powerful 2x2 matrix that dictates a drug's systemic fate by correlating its aqueous solubility with its membrane permeability. Class 1 molecules are the thermodynamic ideal, exhibiting high values for both parameters and often qualify for biowaivers due to their rapid, predictable absorption. In Class 2 and Class 3, we identify specific kinetic bottlenecks. Class 2 is limited by slow dissolution in the gut, while Class 3 is restricted by the molecule's inability to penetrate the lipid bilayer. Class 4 represents the most difficult pharmaceutical scenario, where a substance fails on both fronts, requiring us to solve for both low solubility and low permeability simultaneously. Which of these 4 quadrants would you identify as the primary target for the micronization techniques we discussed previously? To qualify as highly soluble, the highest single dose strength of a drug must fully dissolve in a volume of 250 milliliters or less, a mathematical ratio that must hold true across the entire physiological pH range from 1 to 7.5. This 250 milliliters threshold is critical because it simulates the total fluid volume typically present in the gastrointestinal tract, available for the dissolution process. For the permeability boundary, we utilize a quantitative benchmark of 90%, meaning the drug is only considered highly permeable if 9/10 of the administered dose successfully traverses the biological membranes. If a molecule's solubility fluctuates so much that it requires 500 milliliters to dissolve at pH 1.2, but only 100 milliliters at pH 6.8, how does this failure to meet the boundary across the entire range fundamentally change its BCS classification?
Analyze the upper hemisphere of our biopharmaceuticals classification matrix, where every molecule listed shares the kinetic advantage of high transmembrane permeability. In Class 1, compounds like caffeine and verapamil exist in a thermodynamic ideal, where high solubility ensures they dissolve rapidly in the 250 milliliter gastrointestinal volume, while simultaneously penetrating the lipid bilayer with at least 90% efficiency. Contrast this with Class 2 drugs like ibuprofen and ketoconazole, which maintain high permeability but encounter a significant kinetic bottleneck due to low aqueous solubility. For these Class 2 substances, the absolute rate of systemic absorption is governed strictly by the velocity of the dissolution phase, rather than the membrane crossing itself. Examine this extensive list and identify which molecules would require advanced micronization techniques to overcome their inherent solubility deficits. Analyze the lower hemisphere of our matrix, where every molecule encounters the same kinetic wall, low transmembrane permeability. Class 3 drugs, including metformin and atenolol, satisfy the solubility requirement of dissolving in 250 milliliters or less, yet they fail to achieve 90% absorption because the lipid bilayer acts as a severe flux-limiting barrier. Shift your evaluation to Class 4, where substances like amphotericin B suffer a dual failure. They cannot dissolve effectively in the gastrointestinal fluid, nor can they partition into the biological membrane. This represents the most difficult pharmaceutical scenario because even if you force the drug into solution, the molecule's inherent lack of affinity for the lipid core remains a permanent bottleneck to systemic entry. Looking at these Class 4 examples, what specific structural modifications could you propose to simultaneously lower the dissolution energy barrier and increase membrane flux? Establish the link between the BCS classes and our expectations for in vitro-in vivo correlation by identifying the specific kinetic bottleneck for each category. Because Class 1 molecules possess high solubility and permeability, their systemic arrival is restricted only by the physiological velocity of gastric emptying, making this the primary rate-limiting step.
For Class 2 and Class 3, our enhancement strategies must focus exclusively on the single deficient parameter, either increasing the dissolution rate to liberate the drug from its solid state, or enhancing permeability to overcome the membrane barrier. If you are dealing with a Class 4 substance, you face a compounding kinetic challenge, where systemic availability depends on successfully addressing both low solubility and low permeability within the same formulation architecture.
We are now shifting our focus to the specific engineering strategies used to dismantle the dissolution bottleneck, starting with micronization, which mechanically shears drug particles down to a range of 1 to 10 micrometers. Beyond this, we employ nanonizing to reach a higher order of magnitude in surface area by creating nanocrystals with dimensions as small as 200 to 600 nanometers. Recall the mechanics of the Noyes-Whitney relationship. By drastically decreasing the particle radius, we maximize the surface area available for solvent interaction, which mathematically forces a corresponding spike in the dissolution rate. The final tactic involves the integration of surfactants, which serve a dual purpose as chemical catalysts that lower interfacial tension to promote rapid wetting, while simultaneously modifying membrane fluidity to boost permeability. Examine these three pillars and determine which provides the most significant thermodynamic advantage for a drug that is currently failing due to an extremely low surface-to-volume ratio. Analyze how we can bypass the dissolution bottleneck by converting a drug into its salt form, which effectively lowers the lattice energy and makes the molecules significantly more compatible with the aqueous solvent. By incorporating buffers directly into the formulation, we can control the microenvironment pH, ensuring the drug maintains its optimal solubility profile even when physiological conditions vary. Beyond chemical identity, we must prioritize amorphous and anhydrous fates because their higher free energy and lack of internal water molecules result in a much faster transition into the dissolved state. Based on these three principles, which molecular modification would you prioritize to force a poorly soluble drug into the bloodstream at maximum velocity? Solid dispersions utilize the solvent or co-precipitation method to bypass the limitations of a drug's crystalline structure. We begin by dissolving both the drug and a water-soluble carrier into a common volatile solvent, ensuring both components are mixed at the molecular level. As we apply reduced pressure to evaporate the solvent, the sudden transition prevents the drug from reforming a structured crystal lattice, forcing it instead into a disordered amorphous state. This amorphous precipitate possesses significantly higher internal energy, which translates to a much faster dissolution rate compared to its original crystalline form. Why would the intentional disruption of molecular symmetry through this co-precipitation process be the most effective way to eliminate the lattice energy barrier for poorly soluble compounds? Witness the quantitative superiority of solid dispersions as we plot the concentration of grisofulvin against time for 4 distinct preparations. Observe the lowest curve for micronized grisofulvin. Even with an increased surface area, it fails to reach the high saturation levels achieved by the grisofulvin PVP complexes. As we mathematically increase the ratio of the water-soluble carrier from 1 to 5 to 1 to 20, the dissolution rate, represented by the steepness of the initial slope, and the final plateau concentration both escalate dramatically. This performance occurs because the PVP polymer effectively traps the drug in a high-energy amorphous state, preventing the molecular reaggregation that limits the micronized form. How would you adjust the drug-to-polymer ratio if your goal was to maximize the concentration gradient across the intestinal membrane within the first 30 minutes of administration? The thermodynamic challenge of solid dispersions begins with the solvent paradox, where we must identify a single chemical medium capable of dissolving a hydrophilic carrier and a hydrophobic drug simultaneously. Beyond this molecular hurdle, the resulting material often manifests as a soft, waxy substance that lacks the necessary compressibility and flowability required for industrial tablet production. We must also contend with the instability of the final product, which can lead to non-reproducible solubility and inconsistent dissolution rates across different batches. How would these mechanical flaws, specifically the waxy texture, impact your strategy for maintaining a precise and uniform dose during large-scale manufacturing?
We have successfully integrated the complex interplay between pKa, the partition coefficient, and the dissolution rate to determine a molecule's systemic fate. This synthesis confirms that predicting bioavailability requires a rigorous application of the Biopharmaceuticals Classification System to identify whether solubility or permeability acts as the primary kinetic bottleneck. Given the technical strategies we explored, such as manipulating lattice energy through solid dispersions, how would you now rank these parameters when designing a formulation for a Class 4 compound? Mastery of these molecular mechanics is what allows us to transform a raw chemical entity into a predictable and effective therapeutic agent.